Chapter 2, Sexual Reproduction in Flowering Plants. Summary. Flowers are the seat of sexual reproduction in angiosperms. In the flower, androecium consisting of stamens represents the male reproductive organs, and gynoecium consisting of pistils represent the female reproductive organs. A typical anther is bilobed, dithecous, and tetrasporangiate. Pollen grains develop inside the microsporangia, four wall layers, the epidermis, endothecium, middle layers, and the tapetum surround the microsporangium. Cells of the sporogenous tissue line in the center of the microsporangium undergo meiosis, microsporogenesis to form tetrads of microspores. Individual microspores mature into pollen grains. Pollen grains represent the male gametophytic generation. The pollen grains have a two-layered wall, the outer exine and the inner intine. The exine is made up of sporopollen and anasgium pores. Pollen grains may have two cells, a vegetative cell and a generative cell, or three cells, a vegetative cell and two male gametes at the time of shedding. The pistil has three parts: the stigma, style, and the ovary. Ovules are present in the ovary. The ovules have a stalk called funicle, protective integuments, and an opening called micropyle. The central tissue is the nucellus in which the archaeosporium differentiates. A cell of the archaeosporium, the megaspore mother cell, divides meiotically, and one of the megaspores form the embryo sac, the female gametophyte. The embryo sac is seven-celled and eight nucleate. At the micropylar end is the egg apparatus consisting of two synergid and an egg cell. At the chelazal end are three antipodals. At the center is a large central cell with two polar nuclei. Pollination is the mechanism to transfer pollen grains from the anther to the stigma. Pollinating agents are either abiotic wind and water or biotic animals. Pollen pistil interaction involves all events from the landing of pollen grains on the stigma until the pollen tube enters the embryo sac when the pollen is compatible, or pollen inhibition when the pollen is incompatible. Following compatible pollination, pollen grains germinates on the stigma and the resulting pollen tube grow through the style, enter the ovules, and finally discharges two male gametes in one of the synergids. Angiosperms exhibit double fertilization because two fusion events occur in each embryo sac, namely syngamy and triple fusion. The products of these fusions are the diploid zygote and the diploid primary endosperm nucleus and the primary endosperm cell. Zygote develops into the embryo and the primary endosperm cell forms the endosperm tissue. Formation of endosperm always precedes development of the embryo. The developing embryo passes through different stages such as the proembryo, globular, and heart-shaped stages before maturation. Mature dicotyledonous embryo has two cotyledons and an embryonal axis with epicotyle and hypocotyle. Embryos of monocotyledons have a single cotyledon. After fertilization, ovary develops into fruit and ovules develop into seeds. A phenomenon called apomixis is found in some angiosperms, particularly in grasses. It results in the formation of seeds without fertilization. Apomix have several advantages in horticulture and agriculture. Some angiosperms produce more than one embryo in their seed. This phenomenon is called polyembryony.